Hello, Evan Fahy is my name. I'm here to talk about adipose precursor cell embedded collagen gels and their role in attenuating inflammation and improving tissue perfusion in cutaneous wounds in mice. This work was carried out in Dr. Derek Wan's lab, of which I'm a postdoc. Just as an introduction, cell therapies have been increasing in popularity over the last decade or so, particularly stem cell therapies, due to their potential in regenerative medicine. One area where this might be of benefit is in wound healing. With regard to adipose precursor cells, these are a population that are defined by their adipogenic potential, and we have identified through specific markers a population which exhibits this adipogenic potential, and sought to utilize this to investigate this cell's role in wound healing and indeed what its effects used as a treatment in wound healing may be. With regards to the methods of this paper, we used a standard wound model, which we commonly employ in the lab, whereby the dorsum of mice were wounded with full thickness wounds, bunted open with silicone rings, fastened in place with super glue and sutures to mimic the rate of wound healing seen in humans, with closure by around day 14. The twist that we used in this project was to use nude mice, which could receive cells from another source. In this case, that other source was a Black 6 background ubiquitous GFP mouse, which allowed for green fluorescent cells to be present in these wounds. This meant they were amenable to monitoring throughout, which was carried out with a dermatoscope with UV function. And the wounds were also then harvested for histology, whereby they could be monitored for GFP positivity in this case as well. As you can see with the results, the wound healing rate was rather as expected in the treatment group, or accelerated compared to control, which had not healed by day 14, suggesting this cell population increased the rate of healing relative to control. As well as this, we see that the cells are actually actively taken up into the host environment because there is GFP positivity on histology. GFP mouse skin is shown there just as a reference to the green fluorescence from the donor site, as well as then the transferred cells in situ of the wound tissue border. Furthermore, looking at the cytokine profile of the homogenate from these wound tissues, we find that there is a broadly reduced inflammatory profile amongst the treated cells versus untreated. Additionally to this, laser Doppler scanning was carried out throughout wounding to assess the perfusion of the tissues in treated versus control wounds, and showed that the treated wound maintained a higher level of their perfusion throughout and at day 14 compared to control. So taken collectively, this information shows that the APCs may provide benefit in the context of wound healing, particularly through a reduced inflammatory profile and increased angiogenesis. Whether or not this is direct or indirect is yet to be confirmed and requires further investigation. With regards to the future directions of this work, certainly this cell population deserves more interrogation with respect to the potential angiogenic or anti-inflammatory properties, as well as bioengineering concerns whereby these cells might be altered in vivo to provide a certain fate. Certainly more work to be done and has been underway in the lab, particularly looking at this cell's phenotype at a single cell resolution. With that, I'd like to conclude, and thank you very much for your time and attention.